here. So if we're looking at this, okay? Let me go ahead and take it to full screen. Talk a little bit about this. So here is, just to kind of get an idea, just kind of a refresher. So this happens to be, what is this a diagram of? Tell me, what is that? It's a myofiber, right? Okay? And that myofiber, aka muscle fiber, okay, it's a cell, and it has a membrane. All membranes have all cells have membranes, let's say it that way. All cells have membranes. So in this case, on a myofiber, it has a special name for that membrane. It's called a sarcolemma. So the sarcolemma is going to surround that cell. If it's a cell, it's got to have organelles, a few. It's got to have some content, proteins inside of it. So here we have a myofiber, and you can see that this, on cross-section, the myofiber is divided into myofibrils, which are bundles of myofilaments. Myofilaments is equal to, generically, broad category, actin, thin filaments, and thick filaments, which consists then primarily of actin, which is a protein, and myosin, which is a protein, and then there's an organization to that. So when I say which proteins, you're going to say actin and myosin. When I say thick filament and thin filament, when I say thin filament, I say actin plus troponin plus tropomyosin. That equals one thin filament. If I say thick filament, that means 300 myosin molecules that are bound together. That's a thick filament. So we're going to define it even more. Okay. The same organization you see here on the muscle is what we see on a nerve. So this organization is going to be repeated again on your next unit. Okay, so we have then, this is my axon, and this axon, as you saw on the model, is an extension of a neuron, isn't it? Remember what that neuron looked like? It looked spiny with the body, and then it had this long, looked like a tail coming away. The single tail coming away is the axon. So the information from the neuron, which is part of my nervous system, to bring information to my muscle has got to go down the length of my axon. Now that axon, so there are two cell types that are really excitable. Neurons and myofibers. Those are my two. So that means that they're going to be responding to changes in ion concentrations across the membrane. So we said that those two membranes have what is called a resting membrane potential. So when I say RMP, what does that even mean? It means it's at rest, it's not stimulated. Which ions are greater outside than in? 3 sodium outside to 2 potassium inside, right? That's my standard ratio. We're going to just say that. So at rest, it's going to be 3 sodiums to 2 potassiums across the membrane. When I open the gates, and the gates are voltage regulated, when I open the gate, then that means that, sodium is higher concentration outside than in. So what does it do? Simple diffusion. Rushes in. So it rushes in. And when it rushes in, what happens to the inside of the cell? Does it become more negative or more positive? More positive, right? Because I already have some potassium. Now I've got this bundle of sodium coming in. We call that first part depolarization. So sodium rushes in. And then as it goes in, at resting membrane potential, there is a voltage difference. So at resting, okay, there's a voltage difference. As the sodium rushes in, it becomes more positive inside than out. So there's an electrode that you can put inside in the sarcoplasm, and you can measure that the potential, the charge, goes more positive. So it goes up to positively charged until it reaches a maximum. At that point, we can't do any more. We're done. We shut that gate, and we allow then the sodium to go back in Okay, so what we're going to have is then a shift, and we've got to get back to resting membrane potential. Why do we have to be at resting membrane potential? So it can initiate another action potential, right? It's got to go back to normal. So we close the door, then we can go pass through it again. It's like going through a line. You can only let so many go out, and then you gotta, hey, shut that doggone door and let's start doing it the other way, right? So what you're doing is you're shifting the charge inside the myofiber. That's how the information gets relayed. That's what I was talking about with the wave at the stadium, okay? And that's then the beginning, the initiation where you start the wave, and then when it goes all the way around, that's propagation. Around the entire stadium, that's propagation. Till we get to the end. Then somebody hits another home run, and then all of a sudden another wave goes on, right? You're not doing it all the time. You're doing it when there's something significant that happens. That's the same thing that happens at the sarcolemma. Now, the sarcolemma is here on the myofiber. But down here, it's the same thing. This is an axon, an axolemma, exposed membrane. So it has the same resting membrane potential. Sodium to potassium, okay? So the information's coming down from my brain, through my spinal cord, and it's saying, hey, pick up the 1 pound box of sugar. So I've got to send the message down to my biceps to say, hey, pick it up. So I got to get these muscles to grab, and then I've got to flex my forearm to lift it. Turn my legs and put it on the counter. Okay? All of that information's got to be coming from here. That's why babies can't do that immediately. They figure out how to do that. So what we have then is that the information flow is from the brain to the spinal cord, out the nerve, the motor unit, to then my myofiber. And every myofiber has at least one axon terminal to it, right? It has one. We stated one axon terminal. So we have it coming down here. Now, as this is propagating, when we start doing an action potential, ions are moving. Sodium's moving. Potassium's moving. And so is calcium. So the calcium gates that are only down here are going to open with the voltage change. So the calcium gates open. The calcium that's in higher concentration outside then rushes into that axon terminal. That's where it goes on in. And then when it goes in, the inside of that axon terminal starts to become positively charged, which then in turn activates the various proteins in the axon terminal to allow the vesicles to release the acetylcholine. I need calcium to do that. You remember when we talked about the microtubules and we said that we've got to have the vesicle attach onto the microtubule, and then there's this little engine that kind of pushes it out, gets it to the membrane, and then releases it? Well, it requires calcium. So the calcium has got to come in. It moves then the vesicle to allow for exocytosis. The vesicle in the axon terminal contains acetylcholine. So the acetylcholine gets released. It attaches onto the receptors on this junctional fold, which is actually folded up sarcolemma. The receptors are there. And when it attaches onto the receptor, it then causes a voltage change. It opens the door. Oh, sodium rushes in again, but in this cell. And so we initiate an action potential, that is a depolarization event here, which then is going to ripple out from that point of entry like throwing a pebble in a pond. It's going to go in multiple directions. And we're going to then have an initiation and action potential generated, depolarization. Does that kind of make sense now? Mm-hmm.
Can you explain the chemically voltage channels versus the
voltage gated?
Yeah.
Okay, so when you touch a plug and your hand is by the outlet, what do you feel? Have you ever gotten shocked on a plug?
No.
You don't want to do it. Okay, I've done it. Okay, so you put it in, your fingers are on the stupid probes, and then they go in and you're going, oh, dang. Okay, because there's a shock. That's voltage gated. Chemical means that when you put a chemical that's on a receptor, the chemical is a ligand, which then opens the door. So it's either voltage or chemical, one or the other. So when I'm doing an action potential, that's voltage gated. When I have another chemical that attaches onto it that then opens the gate, that's chemically gated.
So the acetylcholine receptors are the chemical.
Those are the chemical ligands, right?
Yes.
And then that's going to initiate the action potential, which is then going to open up the sodium gates, and that's voltage regulated. That's a good question.
So there's both on that junction, there's both of them.
Oh yeah. On any membrane, period, there's sodium, potassium concentrations on either side. But it's more pronounced on my myofibers and on my neurons. So that's going to then allow for shifts, major shifts in charge, because the sodium is positive, the potassium's positive, and so when I open the door, they all go in. Does that make a little bit more sense in terms of understanding? Took me a long time, trust me, to understand what in the world. Ask me about DNA replication and all the enzymes and topoisomerases and everything else about DNA, I can tell you that. This took me a long time to figure out, okay? How does that go again? And I had to look at pictures because, as I said, I'm a molecular biologist. I'm working at the physiological level, but I'm a microbiologist, molecular biologist. So I didn't work with that, I work with viruses. So it does take time, but all it does is it's looking at videos, 30 minutes, look at them. When something looks overwhelming like a diagram, don't be like me and go, oh my gosh, there's no way. I can't get that one, shut the door. I'll come back to it in half an hour when I feel better. Then you look at it and you go, oh shoot, now I see what's going on. Okay? So you can be alarmed at the first glance, but then take some more time and try to figure it out. Okay, so then when the acetylcholine comes onto this junctional fold to the receptors, it then is going to initiate those changes. Okay, it's going to open the gate, and then the sodium goes in, and then we've got this shift. Okay, so if we're okay with this, I want to keep on going, because I got a lot more to talk about today. Okay, we're okay with that, right? With understanding how that all works. Okay, so if I block the release of acetylcholine, what's the symptom in your patient? If I block the release of acetylcholine, I don't have any contraction, right? It's paralysis. I have to have that attached in order to get contraction. So if I block it, it's not going to go. Okay? If I block, and I can block calcium channels with calmodulin, so I can block it. And so then I cause paralysis. Okay? Now, if I paralyze the activity of acetylcholinesterase, what happens? Yeah, continuous contraction, right? Okay? So what you just need to do is figure out, hey, if I block that, then it can't do that. What does it can't do? That. If I block this, it can't do that, then what's going to happen as a result? So when I have botulism poisoning, then that's going to be a paralysis. Patient's going to die from paralysis of muscles if they're not taken care of. If I have then Clostridium tetani, then that's going to block acetylcholinesterase, and I'm going to get continued contraction throughout my entire body. And that can then cause and result in the death. That's why we have vaccinations. Okay? So, we're okay with this diagram. So we're okay with this too, aren't we? Just tell me, as I move through these slides, are you okay with this one here? So this one here is just showing you the composition. So what's the neuromuscular junction? The axonema, synaptic cleft, and the junctional fold, right, of the sarcolemma. That's my muscular, myofiber junction, where they come together. So what are the structures there? That's what we have. Okay. So we have that pretty well understood. So we're going to go ahead and move along. So then we're gonna, once we get the acetylcholine onto the sarcolemma, we're gonna initiate an action potential. Okay? I'm gonna talk about that. That's where I'm gonna start in a moment. And then any excess acetylcholine that I don't need to pick it up and be contracting in my biceps for the next 30 minutes because I picked up a box of sugar. So I need to get rid of that. I need to stop that contraction. So in order to do that, I have to have acetylcholinesterase. What that acetylcholinesterase does is it just simply take acetylcholine, break it apart, and then they can both be brought back into the axon terminal. The mitochondria are there that make the ATP, that make the bonds, and put it back together. Package it up in the vesicle, and you don't have to make it all over again. Just recycle what you have. So that's what makes it efficient there. Okay, so the neuromuscular junction describes the connection between the nerves and the muscles. Now we know what the components are. The neurotransmitter is going to be acetylcholine. That's going to be really important because when you get over to your 50B, you need to learn dopamine, serotonin, a whole host of other neurotransmitters and what they will do, okay? What causes like Parkinson's, that kind of stuff. So in this case, we're looking at acetylcholine. It's a neurotransmitter. Why do we call it a neurotransmitter? It's a chemical being released by a neuron. That's where it's made. So it's transmitting the information from the nervous system to my muscular system, whether it be skeletal muscle, smooth muscle, wherever it might be. So we have this neurotransmitter, and the acetylcholine is stimulated when calcium goes into the synaptic, the terminal axon, okay? When it goes in there, and then it's released by way of excitosis into the synaptic cleft, aka synaptic gap. So you can see all of that on that model. So don't you think we're going to use that for the lab test? Little white dots for acetylcholine vesicles. Okay? So you need to be able to translate this onto the myofiber model.
Would calcium be a neurotransmitter too?
No. Calcium is just an ion. So neurotransmitter is a very complex protein. Okay, so what happens in an action potential? This is where I was going to at least start, trying to get you into the background on this. So we have to have a resting membrane potential. That means it's not stimulated, it's just hanging loose. What's the charge across the membrane? Well, it's more positive outside than it is inside. Okay, so you can see right here, the brackets represent in chemistry concentration of. So, and also the boldface tells you which one's in higher. That's chemistry punctuation. So the concentration of sodium is going to be higher than potassium. We're still going to have potassium, but it's not going to be the same. They're not equal. And so then on the inside, it's reversed. And then because we have then a greater abundance of the sodium outside than in, this is positively charged here. And we have on the peripheral proteins, they're large molecular weight. They're made up of polypeptides, right? It's a protein. And it's negatively charged. So you've got this huge boulder that's negatively charged. In addition to then, the sodium outside that's being attracted to the negative charge. Opposites attract, right? So it's going to be bundled up toward the membrane. Okay, so the inside face is negative, the outside face is positive. So that's, I'm going to ask you that question. That would be a really good true or false question to start you out on your test. Okay? Intracellular versus extracellular concentration of my ions. Okay. So the major outside ion is sodium, the major inside is potassium. So when we have then an initiation, when this little chemical message comes down here, acetylcholine comes onto here, attaches onto the receptor, then I'm going to start shifting the charge. But I can't do it until those charges begin to shift. So we call that then the initiation of an action potential, and that action potential, if you were to graph it, we'll see the graph of that in a moment, is going to imply then, notice this, just look at this. It goes positively, most of it here, and then you see this section that's negative. And on the inside then, it's positive. So what has happened? Hey, we've reversed the charge. That first part of reversing the charge is called depolarization. So the first part of an action potential is made up of depolarization, followed by repolarization, back to resting. Okay, so I'm going to initiate it's voltage gated, so it's going to cause then the sodium to come on in. And then my action potential is initiated. Now what's going to happen is that I'm going to open my sodium channels. So another good true or false question. Which ion is going to enter during depolarization? I think I had a question very similar to that one. Okay, so it's going to be sodium, right? Okay, so sodium gates open. Okay, so then notice that what's going to happen as I go down this membrane, whether it's a sarcolemma, whether it's an axolemma, I really don't care. It's going to do the same physiology. As it goes down, it moves down segmentally. So this portion is going to undergo the shift. Then when that's finished, it moves down to the next. So we don't go little tiny door, little tiny door. We got a whole building here, and then let's go to the next block, okay? So you're going to have then this regional depolarization. So then again, this is the diagram that's in the lab manual that when I first looked at it, I said, what the heck is this? Okay, well, you kind of look at it and go, oh, okay, I see what's going on here. My synaptic vesicles are migrating to this membrane, and that membrane is my axon terminal. That membrane is actually my axolemma. And inside is going to be my acetylcholine, so it comes over here, and then it begins to flood in that gap. So what causes this to move down here? Calcium. Calcium's going to cause it to move down in here. And so then it releases, and so then when it comes over here, okay, then it's going to attach. Right now, this is showing we just opened the gates. We don't know what's going to happen yet. We just know that we opened the gates. Then on the next one, you can see then that we're going to have this stimulus going down like that pebble in the pond in all directions, all the way. Everything that's membrane is going to now undergo some beginning of an action potential. So you can see that it's going to come down the T-tubules. Okay? And you can see then, here's my T-tubule, there's my terminal cisternae. Those are not going to initiate any action potential. They're just going to only open a gate or close a gate. Open a door or close a door, right? Based upon what happens at the terminal, at the T-tubule. Okay? So the action potential goes down the sarcolemma. The entire myofiber is activated. And so it's in order to get down to the deep parts, because if you look on that 3D model, don't you see some myofibrils in the center? How's it going to get down there? Oh, we only activated the outside? No, the whole myofiber is going to respond. So I got to get it to every single part of that cell. So that means then I'm going to take it all the way down in through between all my myofibrils and activate all my thick and thin filaments. That's what I got to do. That's what I'm trying to do. So once I start this process, once I reach a threshold, I can't reverse it. I can't stop. It's going to go to completion. That's all it's got to do. Okay? So we're going to, it's called, once it reaches threshold, it's all or none. That's what we say. It's all or none once we get to that level. So here is then what's going to happen. So once I have then the sodium channels open, once I reach a maximum, I'm going to close those. I don't want any more sodium coming in. Stop the sodium, And then I'm gonna have to then start diffusing out my potassium. I'm gonna have to get that ratio back up, so I'm gonna have repolarization. So that means then I've got to readjust my charge across the membrane. That's why I have sodium potassium pumps. I gotta assist it. I can't just rely on simple diffusion. I gotta pump it against the concentration gradient to make certain that I get the right ratio of the sodium to the potassium. That process where I close the sodium gates and allow for the potassium to move is called repolarization. So if I looked at it, okay, I'll come back to that in a second. So if I look at it on a chart, just imagine I've got a myofibro. I put an electrode inside of it. And that electrode is going to measure my voltage. Okay? So here I have, if I look at the voltage, I note that at rest, this is resting membrane potential, it's minus 70 millivolts. Okay? That means minus because it's negative inside. Okay, relative to out. That's more sodium outside, right? So it's more negative inside. Once my sodium channels begin to leak, because I get a little bit of leakage, okay, it doesn't, because they're ions and they have a tendency to kind of move anyway. But when I open the door, they're going to rush out. That's no longer leakage. That means I've opened the channel. And at that point, once I get to minus 55 millivolts, that's the measurement, the threshold value that indicates open the doggone doors. That's what we're going to do. We're going to open the doors, and so then sodium is going to rush in. But as it rushes in, it's going to make it more positively charged, right? So it becomes more and more positively charged until I get to the maximum, plus 30. Once I get to that, the cell goes, oh, wait a minute, I can't add any more in here. I'm done. Shut the doggone door. So the door shuts at this point. And then at that point, my potassium channels open. When my potassium channels open, I'm going to start losing some positive charge out, so it becomes more and more negative, right? And then I'm going to sometimes get to this point where I overshoot it. When I overshoot it and I go below that, man, I better clean this up. I better get my sodium potassium pump and get it right. Turn on the pump, get those in the right ratio until I get back to minus 70. Once I get to minus 70, I can go ahead and initiate another action potential. There comes a time, however, when I get to this point here, if I'm on this portion here, that portion there, I cannot stimulate it to open up again. It's called absolute refractory. I cannot open it. I need to shut it, and that's what I've got to do. Okay? So question?
Do you want us to know these numbers for the exam?
You do need to know minus 70.
Okay.
Okay? Because we're getting to the point where we're going to start learning some numbers. Okay. Now notice how long this takes. We're talking milliseconds. It's not this leisurely, let's open the door and let's kind of wait and let's hop on over there. No. We're going, shut, open, shut, close it up. You know. That allows me to initiate another. Let's say that I'm lifting it, but I need to do some more work with that. I got to still take it from the grocery bag over to the counter and maybe into the pantry. So that means I lifted it out. Gonna relax it. Now let's initiate another push. I'm gonna pick it up again and put it into the pantry, right? So I gotta stimulate it again and again. But I can't stimulate it until I get back to resting. It's gotta clean up first. Clean up the sodium and potassium on either side. So if I were to draw something like this on a test, you're able to, and if I labeled it, you're able to tell which part is depolarization, which part is repolarization, okay? Which one is refractory? Where's my threshold? Right? You could put, if I put A, B, C, D or something like that, you could do that. Okay, so depolarization occurs when the sodium ion gates are open. And when acetylcholine then is going to be released, and then we have repolarization when we have sodium ions diffusing. So it's just that graph. You're gonna see that graph everywhere. I don't care. Chemistry, physics, biology, it's the same stinking graph. Okay? Just a different application. Same graph though. Okay, what is the period between depolarization and complete repolarization called? That's called a refractory period. Okay, so this is where I asked you to continue to. So if I have then this action potential being generated, depolarization, it goes down then the T-tubule. The T-tubule is a membrane, isn't it? It's a tubule. And it can then undergo my action potential. These little proteins then, when the action potential comes down here, they're going to activate these proteins, and then that's going to cause my calcium to rush out. Okay? And then the calcium is then going to attach to what? To the troponin. When it attaches to the troponin, what happens to the tropomyosin?
It moves out of the way.
Moves the tropomyosin away from what?
From the active site.
The active site on the actin. What attaches onto that active site?
The myosin.
The myosin. And then when the myosin attaches, okay, now what causes it to attach? Okay, you got to have the ATP, because the myosin head is an ATPase, right? So the ATP attaches at low energy, and then the enzyme becomes active. It splits it, but it doesn't release it. It just holds it in position. When it holds it in position, the myosin heads have enough energy now to attach onto the actin. When it attaches, the ADP and phosphate fall off, and that's when it cranks. Okay? So that then is the next sequence. The action potential is going to be generated. It goes down the sarcomere, the sarcolemma, down into my T-tubule. The best way to do this is if you just label 1, 2, 3, 4, and then give the steps, if you're able to do that. Okay, so then once we get it down on this membrane, it then is going to cause the proteins here to then open these gates, and then the calcium rushes out. So the action potentials triggers the calcium release from the terminal cisternae. Is calcium only found in the terminal cisternae? No, it's the entire SR, right? So I'm going to see it all over in there. And then once that calcium is released, it attaches onto the troponin, moves the tropomyosin, so then that allows the myosin heads to bind to the actin. Forms a cross bridge, okay, just holding on there like a zip line, holds on to that, and then we've got to then do the sliding filament. So then, which protein moves? The actin or the myosin? The actin moves. The myosin is stationary. What keeps the myosin stationary? Yeah, the amyloid. The myomycin in that protein is kind of like a post, and it holds the myosin in place, right? Okay, see, you're catching on. This is good. Okay, so then we have to, when we stop the contraction, we got to pull up all that calcium. We don't want to just waste it and have to generate more. That would be ridiculous. The body is very, very efficient. So the calcium that's released into the sarcoplasm, we got to bring it back into the SR. Right? And start again for the next activation. So this shows, when you looked at this, you probably went, what the heck is that too? Okay, is my sarcoplasm devoid of calcium? No. Calcium is always in there in a low concentration. So some of it's going to bind onto my troponin, right? But I have to have enough of the troponin to be then binding calcium in order for something to happen. I always have background noise. Got to reach threshold. Once I reach threshold, it's going to fire. So what will happen then, in order for that to happen, this is just showing at low concentrations, low intracellular concentrations of calcium. Here's a calcium. This is my little troponin. I've got 3 little parts to it. And so the portion that binds the calcium always has a little bit on there. Again, it kind of falls off and then on there. But it's not enough to trigger anything. So this is then going to be what it looks like when it's in low concentration. So then, in order to have then the tropomyosin come off, I'm gonna have to bind more calcium. When that then binds, it then pulls off my tropomyosin. That requires energy, okay? But tropomyosin moves off, and then that allows then for my myosin heads to attach. So we're always gonna have calcium. Remember we talked about you gotta have between 9 and 11 milligrams per 100 per deciliter of blood. Gotta have to have that. And so that means we always have some hanging around, but in order to actually do something about it, we have to increase the concentration. So then the calcium on the troponin is gonna cause it to change its shape, and then it's gonna lift it. So this is just looking at it from a cross-sectional view. This is not anything but just repeating to you, showing you diagrammatically, hey, it's gonna move that tropomyosin off when it does a conformational change. When the calcium is not there, it looks like this, okay? And it's holding on tropomyosin. When the calcium attaches, it twists and lifts. That's what we call a conformational change. So molecules don't stay static in the cell. They're active and they're inactive. So they're gonna change their shape.
How does the calcium unbind? Or is it just the individual stuff?
No, once you have that coming, it's not a tight bond, okay? It's a transitory. So it will come on and come off, come on, come off. So it's kind of a fluid, but so long as I have most of that area that's now covered on that troponin, on the T and C section, then it will then it will move off. Okay. So then that then explains why we have to have calcium for contraction. I have to remove the tropomyosin so that myosin can attach onto the actin. That's what we call excitation, contraction, coupling. Excitation is, hey, at the neuromuscular junction, when we excite the sarcolemma and we undergo an action potential, down the T-tubule, and we open the doors at the terminal cisternae, that's excitation. Then the release of the calcium, hooking onto the troponin, is the contraction. Excitation, contraction, coupling. They're linked. That's all that's saying. Okay? So we have to have that. So we can have action potential. We can go ahead and take a look at that. Okay. I'm going to continue. I'm going to do a little bit more. Okay. Does it make a bit more sense? Yeah? Okay, so let me go ahead and close this. I should have prepared by opening this up and having it ready to go, but, you know, brain fog. Okay, so then, this is now showing you how that myosin head forms a cross bridge in that sliding filament. A little bit more detail. We were looking at it from the troponin side, okay, tropomyosin. Now let's look to see, but what happens with the myosin heads? So here's my myosin head. In order for it to attach or to at least come up and attach onto the actin active site, I'm going to have ADP and phosphate attached. Okay? So I'm going to have this attaching. Okay? And it will hold on to it for a millisecond. When it comes off, then that's when I'm going to get my power stroke. Now, once it goes to the power stroke, it's used up the energy. It's relaxed. Comes away from that. So I have to have more ATP to then come back in, attach onto the myosin, have it then break it down into ADP and phosphate, attach that ADP and phosphate right there, and then allow it to attach again. So I have to have this continual supply of ATP. In reality, in the body, we only have about 30 seconds worth of storage. of ATP. So if I'm contracting my muscle and we're doing a resistance training, I want to do some weight training. Hey man, I only have 30 seconds of ATP. And I need ATP to do all this. So what does that tell you? In my muscle, I better have some mitochondria. I better be able to make some ATP. And the more I train, the more mitochondria I make. The greater the contraction, the more ATP is available. So when I'm doing resistance training, you can't pick up 300 pounds the first day that you go in there. I mean, you try. You look like an idiot in the gym. But you have to kind of build it up over time. And as you build it up, what you're doing is you're building up the muscle and making more mitochondria. So that over the course of time, you're able to lift more weights without fatigue. Remember, skeletal muscle fatigues really quickly. And the only way to overcome that is energy, oxygen, and fuel. That's it. So that takes time to kind of develop that. Okay, question?
And where's the mitochondria?
Okay, good question. All right, so so where are mitochondria usually located in any animal cell? In the cytoplasm of the cell, right? What's my cell in my muscle? Within the sarcolemma, right? So I have myofibrils. They're so jam-packed with bundles of actin, thick and thin filaments, I can't get any mitochondria in there. But in between my myofibrils, I got lots of room to make more mitochondria. So the mitochondria are inside the myofiber because it's a cell. Mitochondria is an organelle. It's got to be in a cell. But it can't be in a myofibril because I've got bundles of all kinds of thick and thin filaments. So they're just right outside of it. So look at your myofiber model, and you can see the mitochondria on there. Okay? Okay. So, continuing on. So the way in which I'm going to have then, here I have then my ATP. My myosin head is relaxed, and now it's exposed. My substrate, ATP, can attach. The attachment then activates this enzyme to break it down and hydrolyze it into ADP and phosphate. Just break the bond, right? But they're still connected, so it makes it now able to then attach. Once it attaches, it causes the ADP and phosphate to fall off, and then that causes the power stroke. So that would be a really good test question. Which of the following conditions results in the power stroke of a myosin against an actin? Okay? So that's what this particular diagram is trying to show you. And we keep doing that, keep doing it until there's no more calcium. When there's no more calcium, okay, or no ATP. If I don't have any ATP, that's what we call when you're on a marathon and you haven't trained enough and you've used up all your fuel and you have no more ATP and you've hit the wall, you can't do anything more. Someone's going to have to carry you the rest of the way or pick you up somehow. Okay? Because what you're trying to do when you're training for a marathon is, again, increase the number of mitochondria, supply then more glycosomes with the ability to form glycogen stores. The more glycogen stores, the more readily available the substrate is for glycolytic pathway. And that does not happen overnight. So when you're training for a marathon, what is it, 6 months, 3 months? You won't catch me out there doing that. I'll run a 5K, but I don't know. I just can't do that. Okay? It's a lot of work. And you have to be able to then have an endurance to get to that point, and you got to train to do that. Okay. If I don't have, okay, if let's say, for example, forensic pathologists, when they go out into, they find a body and they're trying to figure out, well, how long ago did this person die? You know, because they've got to determine the time of death perhaps when they're doing an investigation. And they may notice the relative stiffness, the rigor mortis. And there is a a diffusion of calcium away from the site, and then the body becomes more flaccid. So initially, it's gonna be contraction. You're gonna get rigor mortis, all the calcium just kind of comes out of everywhere. All of the SR just spills out all the calcium. It then attaches onto all of the myosin heads, onto the troponin, and you get the contraction. And that will then continue until all the calcium is bound. Then it's gonna slowly diffuse away because it's a transitory bonding. The calcium onto the troponin. So then as it goes away, then they can tell how long that body, from that point on, from the point of rigor mortis to being flaccid, how long that body has been there. Have you ever read The Body Farm? Oh man, you gotta read it. I love detective novels. Patricia Cornwell, she wrote The Body Farm. She was a police investigator in her real life, became a novelist, and The Body Farm is a study at University of Tennessee where they actually have, it's called The Body Farm, they put bodies in various states of decay, like under the soil, in a shed, and just right on the dirt, 6 feet under, so that what they do is they look at how the body decomposes. Which insects come first? Which types of parasites go in? And then once you go through and you monitor, how hot was it? How cold was it? You can actually determine a lot of stuff. That's why the FBI trained there. Because it's a body farm. You can actually do these kinds of things with bodies, Somebody's got to say, yeah, go ahead and take my body, go ahead and do that. But it's a true to life. That's how they actually determine the length of time. But it's a fiction. Okay? But it's a real I like this kind of sick things. Okay, so we then have, that's why I like to watch all these medical shows. I like to watch, what was it, the Dexter, remember when Dexter was on? Oh, I love Dexter too. I wish, so there's a lot of crazy things that I like to watch. Okay. All right, so are we okay with this so far? Okay, I think I did a really good overview of this. If you weren't listening, I hope you taped it because, you know what? I went over everything on this. So then tomorrow, on Thursday, I'll go ahead and do the next 2 sections. I'm going to go ahead and start with muscles and talk about aerobic, how muscles form energy with ATP. We have to have ATP, right? Because, and the thing about muscles is that I might want to have a quick contraction. I might want to have a slow contraction. I might want to have a 1 pound weight. The force of contraction can be variable, and the rate of contraction can be variable. So I've got to look at it now. We know what the cellular level is. What happens at the muscle, at the organ level? Remember, every muscle in your body is its own separate organ. So how does it contract, and how does it then make the ATP? Because you saw, I need calcium. Now we know why. Got all that information about calcium homeostasis. Okay? I need to have fuel to make ATP. Anybody recall glycolysis was my substrate? How about glucose? But in my body, in my skeletal muscles, I form glycogen. Glycogen is a polysaccharide of glucose. So I have to store my glycogen in my muscle to provide the glucose to go into ATP production. So tomorrow Thursday we'll talk about how the muscle makes ATP energy. So if you need to, you bring your tape recorder, you bring your phone, and you tape it. Okay? And then we'll finish it out on Tuesday because on Thursday we have a test. Okay? And I'll be generous. I won't give another quiz. I will give you a review quiz just for you to look at on this next section. But I'm going to give you on Tuesday. On Tuesday, a group assignment on all the muscles. I'm going to send it just to the biceps brachialis. Give the bony attachment, the origin for the iliopsoas. right? So we will be done with muscles this week. You get the weekend to kind of put it together, and then on Tuesday, we'll have a group assignment on the muscles. Okay, lots more to do, but hey, we're okay. All right, so let's go ahead and we'll start lab. First thing you want to do when you go to lab is you want to review the muscles that you've already studied. Head muscles, neck muscles, shoulder muscles, back, chest. Go make certain you know all those. The origin of the insertions. And then by the end of tonight, you need to be done with arm, the whole upper, chest, back, and hip. I need you to get that done. Because Thursday, we've got to finish all the muscles of the thigh and the lower leg. So that's what we're going to gauge it. I need everything.